The biotechnology principles and process. The biotechnology deals with the techniques of using the life organisms from organisms to products and processes useful to humans, and the aim is sense of making God and the bread or wine, which are all the micro-mediated process that could be thought as a form of the biotechnology. However, in a restricted sense, and that will be referred to as a social organism, which is a genetically modified organism to achieve the same digestion and the further many other techniques are under biotechnology. And for example, in the population of the living tissue, using synthetic energy and using and developing energy and the correcting and efficiency are all part of biotechnology. The definition of biotechnology, we have given a definition of biotechnology that encompasses both the traditional view and the modern molecular biology. Biotechnology and the definition given by ESG for the division of the natural size and cells for data from the molecular analogs of products and services. The principle of biotechnology, among many, the two core techniques that enable the both of the modern biotechnology are the genetic engineering, the techniques to alter the chemistry of the genetic material, the DNA and RNA, and to produce the host organism, and the change the phenotype of the host organism. The work of engineering and maintenance of the cells, microbial contamination, and chemical engineering processes to enable the growth of only the desired microbe or eater itself in large quantities for the manufacture of the biological products like antibiotics, vaccine, enzymes, etc. Let us now look at the conceptual development of the principles of genetic engineering. It will appreciate the advantage of the sexual evolution over asexual evolution. The former provides opportunities for variation and formulation of the unique combinations of genetic setup, and the sum of which may be beneficial to the organism as well as to evolution. The sexual evolution and the preserve the genetic information by sexual reproduction. And this is because the linked base of the DNA has become a part of the chromosome and which has ability to replicate, and nine chromosome there is a specific DNA sequence of the original replication which is essential for initiating replication. Therefore, for a multiplication of an alien piece of DNA in an organism, it needs to be a part of the chromosome which has a specific sequence known as the original replication. Thus, an alien DNA is linked with the original replication so that the alien piece of DNA can replicate and multiply itself in the host organism. And this can be called as the cloning or making multiple copies of any template DNA. And later focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene and encoding antibiotic resistance with a native plasmid organism for replicating cellular extracellular DNA. Self-analyzed Newman and recently Cohen and Harvard were accomplished this in 1972 isolating the antibiotic resistance gene by cutting out a piece of DNA from plasmid, and which was responsible for conferring the antibiotic resistance. The cutting of the DNA specific location became possible with the discovery of the so-called molecular accesses, the restriction enzymes. The cut piece of the DNA was then linked with the plasmid DNA, and these plasmid DNA act as vector to transfer the piece of DNA attached to it. And you probably know that the mosquito acts as an insect vector to transfer the mineral parasite into the human body. In the same way, the plasmid can be used as a vector to deliver an alien piece of DNA into the host organism, and the linking of the antibiotic resistance gene with the plasmid vector became possible with the enzyme, the DNA ligase, which acts on the cut DNA molecules and joins their end. Then this makes the new combination of the circular organisms replicating DNA in the ingredient is known as recombinant DNA, and when the DNA is transferred into Escherichia coli or bacterium from protein to the salmonella, it could replicate using the new host bacteria, new host DNA polymerase enzyme and make multiple copies, and this ability to multiply copies of the antibiotic resistance gene in E. coli was called the cloning of antibiotic resistance gene in E. coli. You can hence infer that there are three basic steps in the genetically modifying organism, and the identification of the DNA with desirable genes, and the introduction of the identified DNA into the host, and the maintenance of the introduced DNA in the host, and transfer of the DNA to the DNA, and the tools of the recombinant DNA technology. Now we know from the following discussion that the genetic engineering of the recombinant DNA technology can be accomplished only. We have the key tools in the restriction enzyme, the polymerase enzyme, ligase, vectors, and the host organism, and the let us try to understand some of these in detail. The restriction enzymes in the year 1963, the two enzymes responsible for restricting the growth of the macrophage in Escherichia coli was isolated, and one of these added methyl groups to DNA while the other cut DNA, and the latter was called the restriction endonuclease. The first restriction endonuclease gene whose functioning depended on a specific DNA nucleotide sequence was isolated and characterized. 5 years later, it was found that the hill to always cut DNA molecules are as particularly defined by recognizing the specific sequence of sequence, and this specific base sequence is known as the recognition sequence of the Hindu, and besides the Hindu, we know more than 900 restriction enzymes there have been included along with 20 systems of vectors, and each of which recognize different recognition sequences. In the convention for naming these enzymes is the first letter of the name comes from the genus, and the second letter comes from the species of the prokaryotic cell from which they were isolated. For example, the Escherichia coli comes from the Escherichia coli, or third Indigo or one, or is derived from the name of The string the Roman numbers following the names indicate the order in which the restriction enzyme isolated from their chain of bacteria. The restriction enzyme belong to larger class of enzyme called the nucleus, and these are the two points there is a nucleus and the nucleus. This nucleus removes the nucleotide from the ends of the DNA sequence overall, and the nucleus makes what's the specific position within the DNA, and each restriction and the nucleus function by inspecting the length of a DNA sequence, and once it finds a specific recognition sequence, and it will bind to the DNA and cut each of the two strands of the double helix in a specific points at the sugar phosphate backbones, and each restriction in the nucleus recognizes a specific palindromic nucleotide sequence in the DNA. The restriction in the nucleus cut the strand a little away from the center of the palindromic site, but between the same two bases of the opposite strand, and this leaves the single stranded portion at the ends, and there are overhanging stretches called the sticky ends on each strand, and they are named so because they form the hydrogen bond with the complementary counterpart, and the stickiness of the ends facilitates the action of the enzyme DNA ligase. The restriction in the nucleus are used in gene to form the recombinant molecules of DNA, which are composed of the DNA from different sources or genomes. When cut by the same restriction enzyme, there are overhanging stretches called the sticky ends on each strand, and they are named so because they form the hydrogen bonds with their complementary counterparts, and the stickiness of the ends facilitates the action of the enzyme DNA ligase. The restriction in the nucleus are using the anti-DNA to form the recombinant molecules of DNA, which are composed of the DNA from different sources or genomes. When cut by the same restriction enzyme, the resulting DNA fragments have the same kind of sequence, and thus can be joined together into using the DNA ligase. In the restriction nucleus are used to form recombinant molecules of DNA, which are composed of the DNA from different sources or genomes. When cut by the same restriction enzyme, the resulting DNA fragments have the same kind of sticky ends, and this can be joined together into using the DNA ligase. You may have realized normally unless one cuts the vector and the source DNA with the same restriction enzyme, the reduction vector molecule cannot be created. And the separation and the isolation of the DNA fragment, the cutting of the DNA restriction in the results in the fragments of the DNA. These fragments can be separated by technique known as the electrophoresis. The DNA fragments are negatively charged molecules, and they can be separated by forcing them to move towards the node. Combined electric fields through medium or matrix, and nowadays the most commonly used matrix is agarose, which is a natural polymer extracted from the seaweed. Then the DNA fragments separate or resolve according to the size through the sieving effect provided by the agarose gel, and hence the smaller fragment size and the further it moves. And the separate DNA fragments can be visualized only after staining the DNA with a compound known as NCERT bromide, followed by fluorescence UV and mineralization, and you cannot see the pure DNA fragment in the visible light, and without staining, and you can see the bright color arrangements of DNA in Ethereum bromide strain gel being exposed to UV light. The separated bands of DNA are covered from the agarose gel and extracted from the gel using this step is known as solution, and the DNA fragments purified in the way are used in constructing the recombinant DNA by joining them with the cloning vectors. You know that the plasmid and the vector phase have the ability to replicate within the vector cell independent of the control of the chromosomal DNA vector phase because of the high number per cell have very high copy numbers of the genome in the vector cell, and the some plasmid have only 1 or 2 copies per cell, and whereas the others may have the 15 to 100 copies per cell, and their number can go even higher. And if you are able to link and link bits of DNA within vector phase or plasmid, then we can multiply its numbers equal to the copy number of the plasmid or vector, and vector is used as agent of engineering such a way that this helps in the linking of the foreign DNA and selection of the recombinants from non-recombinants. And the following are the features that are required to facilitate the cloning into a vector. In the original reduction provided in the sequence from where the original reduction starts, and any piece of DNA when linked to this sequence can be imaged typically within the host cells, and this sequence is also responsible for controlling the copy number of relevant DNA. So if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin supports high copy number, and the selectable marker in addition to arrive the vector requires a selectable marker which has been identified, eliminating the non-transformation and selectively permitting the growth of the transform. Transformation is the procedure through which a result is introduced in a whole vector, and eventually the process in a subsequent section. And normally the genes encoding the resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, akanamycin, etc. are considered useful selectable markers for E. coli, and the normal E. coli cell do not carry the resistance against any of these antibiotics, and the cloning sites in order to link the alien DNA. The vector need to have a very few, preferably single recognition sites for the commonly used restriction enzyme, and the preference of more than one restriction site within the vector will generate the several fragments, and which will complicate the gene cloning and the ligation of the alien DNA is carried out at the restriction site and present in one of the two antibiotic resistance gene. For example, you can ligate a foreign DNA at the one site of the tetracycline resistance gene. In the vector PBR322, and the recombinant plasmids will lose the tetracycline resistance due to the insertion of the foreign DNA, but can still be selected out from the non-recombinant one by plating the transformants on the tetracycline containing medium. The transformant going on the ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinants will go in the ampicillin containing medium but not on that containing tetracycline, but the non-recombinant will go on the medium containing both the antibiotics. And in this case, the antibiotic resistance gene helps in selecting the transformants. The transformant are going on the ampicillin containing medium and then transformant medium containing tetracycline. The recombinant will go on the ampicillin containing medium but not on that containing tetracycline, but the non-recombinant will go on medium containing both the antibiotics. In this case, the antibiotic resistance gene helps in selecting the transformants, while the other antibiotic resistance gene gets inactivated due to insertion of the alien DNA and helps in selection of the recombinant and the selection of the recombinant due to the inactivation of the antibiotic. In the combustion process, and because the simultaneous splitting on the two players having the different antibiotics, and therefore the alternative selectable markers have been developed which differentiate the recombinant from the non-recombinant on the basis of the ability to produce color in the presence of the recombinant substrate. In this, recombinant DNA is inserted with the coding sequence of the enzyme in the beta collector site, and there is due to inactivation of the enzyme. The beta collector site is this results into inactivation of the gene for the synthesis of the enzyme, which is referred to as the insertional inactivation. The presence of protein substrate in the gene, the blue color colonies of the plasmid in the bacteria does not have an enzyme, and the presence of the enzyme results into the insertional inactivation of the beta collector site gene. In the cloning, and the colonies do not produce any color, and they are identified as the recombinant colonies in the vectors for the cloning gene. In plants and animals, you may be surprised to know that we have learned the lesson of transferring genes into plants and animals and bacteria and viruses, which have known this for ages, and how to deliver the genes to transform the eukaryotic cells and force them to do what the vector or the viruses want. And for example, the vector interactions. The president of the several type of plant is able to deliver a piece of DNA of the TDM to transform the normal plant cells into a tumor, and this human cells to produce the chemicals required by the pathogen. And similarly, the retroviruses animal cells have the ability to transform the normal cells into cancerous cells. A better understanding of the delivering genes, the tumor-inducing plasmid of the agrivector innovations has now been modified into cloning vector, which is no more pathogenic to the plant, but is still able to use the mechanisms to deliver the genes of our interest variety of plants. And the similar retroviruses have also been designed and are now used to deliver the desirable genes in the animal cells. So once gene or DNA fragment has been aerated into cerebral vector, it is transformed into a bacterial, plant or animal host, and the various multiple component host. And this DNA is a hydrophilic molecule. It cannot pass through the cell membranes. While in order to force the vector to break up the plasmid, the vector cell must first be made competent to take up DNA, and this is done by treating them with a specific concentration of divalent cation and such as the calcium, which decreases the affinity of the affinity with the DNA enters the vector through the pores in cell wall, and the recombinant DNA can then be forced into such cells by incubating. The cells with the recombinant DNA are nice, and followed by placing them briefly at the 42 degrees centigrade heat shock, and then putting them back on ice and the reduction of the shock. Break up the recombinant DNA, and this is not only the way to introduce the alien DNA into host cells in a method known as a microinjection. Recombinant DNA is directly injected in the nucleus of an animal cell. In another method suitable for plant, the cells are bombarded with high velocity microparticles of the gold or on the core with the DNA in a method known as the ballistic charging gun. And the last method uses the design vector in which are allowed to infect the cell, transfer the recombinant DNA into the host. Now that we have learned about the tools for constructing the recombinant DNA, the process of the recombinant DNA technology and the recombinant DNA technology involve the several steps in specific sequence, such as isolation of the DNA and fragmentation of DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, cutting the host cells in the medium at a large scale, and extraction of the desired product. And the later examine each of these steps in some details. The isolation of the genetic material DNA, and the recall that the nucleic acid is the genetic material of our organism without exception. In majority of the organism, this is deoxyribonucleic acid or DNA. In order to cut the DNA with the restriction enzymes, it needs to be in pure form, free from other macromolecules. In the sense the DNA is enclosed within the membrane, and we have to break the cell open to release the DNA along with other monosaccharides to break. So These are the molecular molecules such as RNA, protein, polysaccharides, and also lipids. And this can be achieved by reading the vector cells. Animal or plant tissue with enzymes such as the lysosome, bacteria, and the cellulose plant cells and the chitinase fungus. And you know that the genes are on long molecules of the DNA intervene proteins such as the histones and the RNA can be removed by treatment with the ribonuclease, where the proteins can be removed by treatment with the protease, and the other molecules can be removed by the appropriate treatments and purified. DNA ultimately precipitates out after the addition of the fuel ethanol, and this can be seen as a collection of the fine threads in suspension. The cutting of DNA has specific locations. The restriction enzyme reactions are performed by incubating the purified DNA molecule with the restriction enzyme at the optimal condition for that specific enzyme. In the aggressive electrophoresis is employed to check the progression of a restriction enzyme reaction. The DNA is the negatively charged molecule, hence it moves towards the positively charged electrode anode, and the process is repeated with the vector DNA also. The joining of the DNA involves several processes after having cut the source DNA as well as the vector DNA with the restriction enzyme in the DNA is enough interest on the source DNA and the cut vector. This base and means and ligase is added in the results in the revelation of the DNA and amplification of the DNA center using the picture in the PC strands of the polymer chain. This is the multiple copies of the gene for DNA of interest is synthesized in vitro using two sets of primers in the small chemically synthesized oligonucleotide. They are complementary to the origins of the DNA and the DNA polymerase enzyme extends the primers using the nucleotide provided in the region, and the genomic DNA is tempered with the process of the replication of DNA is repeated many times. The segment of the DNA can be amplified to approximately billion times, and the 1 billion copies are made. Such repeated amplification is achieved by the use of a thermostable DNA polymerase isolated from bacterium. The thermocyclotron which remain active during the high temperature induce the denaturation of the represented DNA. The amplified fragment of the desired can now be used to ligate with the vectors for further cloning and the insertion of the recombinant DNA into the host cellular organism. There are several methods of introducing the ligated DNA into recipient cells, and the recipient cells, after making them competent to receive, take up for instance into an antibiotic. Example, ampicillin is transferred to E. coli in the cells, and the host cells become transformed into ampicillin resistant cells. If we spare the transformed cells on a goblet containing ampicillin, only transformants will go on the untransformed recipient cells will die in the cells due to ampicillin resistant gene. The one is able to select a transformed cell in the ampicillin resistant gene. In this case, is called the selector model of obtaining the foreign gene product. When you insert a piece of alien piece of DNA in the cloning vector, insert into bacterial plant animal DNA, alien DNA get multiplied in almost all the recombinant DNA nucleases. The ultimate aim is to produce the desirable gene, and the historical is for the recombinant DNA to be expressed and the foreign gene get expressed under appropriate condition. The expression of the foreign genes in the host cells involves the understanding many technical details after having cloned the gene of interest, having optimized the condition to induce the evolution of the protein. One is to consider producing it on a large scale. Can you think of any reason why there is a need for the large scale production if any protein encoding gene is expressed in a heterogeneous source, and it is called recombinant protein, and the source of the clone genes of interest may be grown on a small scale in the laboratory. The source of the clone genes of interest may be grown on a small scale in the laboratory, and the cultures may be used for extracting the DNA, extracting the desired protein, and then purifying it by using the different recognition, the different separation techniques, and the source can be utilized in the continuous culture system as a variant. The use medium is gained from one side to side on the world. Fresh medium is derived from another. Derived from the other to maintain the cells in the physiologically most active log or exponential phase, and this type of the culturing method reduces the large minus leading to the higher range of the desired protein. The small volumes of the culture cannot yield appreciable quantities of products to produce in large quantities. The development of the bioreactors, a large volume, that is the 100 to 1000 liters of the culture can be processed was required, and thus the bioreactors are thought of as vessels in which the raw materials are biologically converted into specific products and the individual enzymes, etc. using the microbial plant, animal or human cells. The bioreactor provides the optimal condition for achieving the desired product by providing the optimal growth conditions, the temperature, pH, substrate, salt, vitamin, substance. The most commonly used bioreactors are of the styrene type. A stirred tank reactor is usually cylindrical or with a curved base to facilitate rinsing out the nuclear reactor contents. The toxin availability throughout the various and ultimately used in the regulatory system and oxygen delivery system and the foam control system and the temperature control system, the pH control system, and sampling port that a small volume of the culture can be withdrawn theoretically and the downstream processing after completion of the bioenzymatic stage, and the product has to be processed through series of the process before it is ready for marketing. And the finished product, the process includes the separation of the process includes the separation and purification of the which are collectively referred to as the downstream processing, and the purification has to be included with the certain drivers. Suitable preservative in the suspension has to undergo the clinical trial. As in case of drugs, and the strictly quality control testing for each product is also required, and the downstream processing and quality control testing and the vary from product to product. Biotechnology and applications. Biotechnology essentially using the patient will include the therapeutic diagnosis and genetically modified crop for agriculture, processed food, bioremediation, the waste treatment, and energy production. The three critical research areas are providing the best catalyst in the form of the improved organism, casually a microbe or pure enzyme, and creating optimal conditions through engineering for catalyst to act in downstream processing technologies to purify the protein or organic compound, and biotechnological application in agriculture. Let us look at the three options that can be for increasing the food production and the agrochemical based agriculture. Organic agriculture and the genetically engineered crop based agriculture. The Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population, and the increased yields has partly been due to the use of the improved crop varieties, but mainly due to the use of the better management practices and use of the agrochemicals, fertilizers and pesticides. And however, for farmers in the living world, the agrochemicals are often too expensive, and further increase in yield with existing varieties are not possible using the conventional breeding. Is there any alternative path that our understanding of the genetics can show so that any farmers can obtain the maximum yield in their fields? And is there a way to minimize the use of the fertilizers and chemicals so that the harmful effects on the environment are reduced, and the use of the genetically modified crop is a possible solution? Plant bacteria, fungi, and animals whose genes have been altered by the manipulation are called the genetically modified organism, and the GM plants have been used in many ways, and the genetic modification have made crops more tolerant to abiotic stresses on the cold, drought, salt, heat, and the reduced reliance on the chemical pesticides or pest-resistant crops, and they help to reduce the post-harvest losses, and they increase the efficiency of the mineral usage by plants, and this prevents the early exhaustion of the fertility of soil, and the enhanced nutritional value of food. Example, the golden rice, that is vitamin A in rich rice. In addition to these uses, and the GM has been used to create the tailor-made plans to supply the alternative resources to industries in the form of the starches, fuels, pharmaceuticals, and some of the applications of the biotechnology in agriculture that you will study in detail, or the production of the proposed pest-resistant plants, and which could decrease the amount of the pesticide use. And the berry-toxin is produced by the bacterium called the Bacillus thuringiensis, and the berry for short. And the berry-toxin gene has been grown from the bacteria and can express in plant to provide the resistance to insect without the need of for insecticide in effect created by pesticide. And the examples are the berry corn, rice, tomato, potato, and soybean, exception. The berry corn and some of the things are the such as the lepidopteran, the tobacco bottom, and the armyworm, coleoptera, beetles, dipterans, flying mosquito, and the Bacillus thuringiensis from protein crystals that form a particular phase of the growth, and these crystals contain a toxic insecticidal protein. And why does this toxin not kill the bacillus? Actually, the berry toxin protein exists as inactive toxin, and once an insect agent ingests the inactive toxin, it is converted into the active form of the toxin due to the alkaline pH of the gut, which solubilizes the crystals, and the activated toxin binds to the surface of the midgut epithelial cells and create pores that cause nasal swelling and lysis, and eventually cause death of the insect. And the toxin genes are isolated from the Bacillus thuringiensis and incorporated into several cropland from the cropland such as cotton. And the choice of the genes and depend upon the crop. And the target pest as the most berry-toxin are the insect group specific. The toxin is encoded by a gene. The toxin coded by a gene Cry1AC named Cry, and hence the number of them. For example, the proteins encoded by the gene the Cry1AC and Cry2AB control the cotton bollworm, and that of the Cry1AB controls the corn borer. In the pest resistant plant, the several nematodes and the parasite parasitize in the wide variety of the plants and animals, including human beings, and emit the melodica and incognitia in the infect the root of the tobacco plants and cause the great reduction in yield. And the novel strategy was adopted to prevent this infection, which was based on the process of the RNA interference, RNAi, and the RNAi takes place in all eukaryotic organisms as a method of the cellular defense. And this method involves the silencing of a specific mRNA due to complementary double-stranded RNA molecule that binds and prevents translation of the mRNA silencing. And the source of this complementary RNA could be from an infection by viruses having the RNA genomes or mobile genetic elements in the transposons and viruses transposons in the liquid via mRNA intermediate using vectors. The nematode-specific genes were introduced into the host plant. The introduction of the DNA was such that it produced the both sense and antisense RNA in the host cells, and these two RNA being complementary to each other in the form of double-stranded DNA. DNA then initiated the RNAi and thus silenced the specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing the interfering RNA, and the transgenic plant therefore got itself protected from the parasite. And the biotechnological application in medicine and the recombinant DNA technological products have made the immense impact in the area of the healthcare by enabling the mass production of the safe and more effective therapeutic drugs. And further, the recombinant therapeutics do not. The biotechnological application in the medicine and recombinant DNA biological processes have made the immense impact in the area of the healthcare and by enabling the mass production of safe and more therapeutic drugs. And further, the recombinant therapeutics do not induce unwanted immunological responses as is common in case of the similar products isolated from the non-human sources. As at present, about 30 recombinant therapeutics have been approved for human use the world over in India, and 12 of these presently being marketed. And the genetically engineered insulin and the management of the adult onset diabetes is possible by taking insulin at regular time intervals. And what would be every patient do if enough human insulin was not available? If we discuss this, we would soon realize that one would have to isolate and use insulin from other animals, and what the insulin from the other animals is just regulated by the human body itself. And what we and so now imagine if the bacteria were responsible that could make the human insulin suddenly the whole process and it becomes as simple, and they can easily grow large into the bacteria and make more insulin as you need. And think about whether insulin can be transferred in every patient or not, then it could be digested. The insulin used for diabetes were earlier extracted from the pancreas of the slaughtered cattle and pigs, and the insulin from an animal source, and though cause some patients to develop allergy or other type of the reaction to the foreign protein. And the insulin consists of the two short polypeptide chains, the chain A and chain B, that are linked together by disulfide bridges in mammals and including humans. And the insulin is synthesized as a pro-hormone like a pro-enzyme, and the pro-hormone need to be processed before it becomes a fully mature and the functional hormone, and contains a extra stage called the C peptide, and this peptide is not present in the mature insulin and is removed during the maturation into insulin. The main challenge for the production of the insulin during the RNA or DNA techniques was getting insulin assembled to a mature form in 1983, and the originally an American company prepared two DNA sequences corresponding to A and B chains of the human insulin and introduced them in plasmids of E. coli to produce the human insulin. The chain A and chain B were produced separately and extracted and combined by creating the disulfide bond to form the human insulin. The gene therapy, if a person is born with hereditary disease, and can a corrective therapy can be taken for such a disease, and the gene therapy is ideal to do this. And the gene therapy is a collection of the methods that allows the correction of a gene defect that has been diagnosed in a child or embryo, and here the genes are inserted into a person's cells and tissues to treat the disease. And the correction of a genetic defect involves the delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. And the first clinical gene therapy was given in the 1990s to a 4-year-old girl with the adenosine deaminase deficiency. The enzyme is crucial for the immune system to function. That is always caused by the deletion of the gene for adenosine deaminase. In addition, in some children, the deficiency can be cured by bone marrow transplantation. In others, it can be treated by the enzyme replacement therapy and increase the function. ADA is given to the patient by injection. The problem with both of these approaches that they are not completely curative, as the first step towards the gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body of function like here. A feed using a retroviral vector is then introduced into this lymphocyte, which are subsequently returned to the patient. And however, as these cells are not immortal, the patient requires the periodic infusion of such genetically engineered lymphocytes. And however, if the gene is isolated from the marrow cells and the producing the ADA is introduced into the cell membrane stage, it will be a permanent cure. The molecular diagnosis. For the effective treatment of the early diagnosis and understanding the pathology is very important. The conventional methods or issues of the ceremony diagnosis in the early reaction is not possible in the biotechnology. Polymerase chain reaction, PCR, enzyme link, immunosorbent, these are some of the techniques that allow the purpose of the early diagnosis. Presence of the pathogen bacteria, virus, etc. normally suspected only when the pathogen is produced. Then has produced a disease symptom, and by this time in the concentration of the pathogen is already very high in the body and however very low concentration of the bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected. The implication of the nucleic acid by PCR, and they can explain how PCR can detect the very low amounts of the DNA. The PCR is now routinely used to detect the HIV in suspected cancer patients in the powerful individuals. The PCR is now routinely used to detect the HIV in suspected AIDS patients, and it is being used to detect the mutation induced in suspected cancer patients too. It is a powerful technique to identify many other genetic disorders. Single-stranded DNA tag with reactive molecule of probe is allowed to hybridize into complementary DNA in the clone cells. In a clone of cells and the photographic detection using auto-radiography, the clone having the mutated gene will hence not appear in the photographic film, and because the probe will not have the complementarity with the mutated gene, the leak is based on the principle of the antigen-antibody interaction, and the infection by the patient can be detected by the presence of antigens suppressing glycoprotein, etc., or by detecting the antibody synthesis against the pathogen. In the transgenic animals that have the DNA manipulated to process and express an extra foreign gene, and non transgenic animals, transgenic rats, and the rabbits, fish, sheep, cows, and fish have been produced. Although 95% of all existing transgenic animals are mice, and normal physiology and development in the transgenic animals can be specially designed to allow the study of how the genes can be regulated and how they affect the normal function of the body and its development. Resemble the studying of the complex factor involved in growth, such as the insulin-like growth factor, and by introducing the genes from other species that alter the formation of this factor and studying the biological effects of the result. The information is obtained about the biological role of the factor in the body and the study of the disease. And the many transgenic animals are designed to increase our understanding of how the genes contribute to the development of disease, and the disease especially made to serve as models for human diseases. Certain restriction of the new treatments of all diseases is made possible in the transgenic model exists for the human diseases, the cancer, cystic fibrosis, the rheumatoid arthritis, and Alzheimer's, and the biological products. The medicine are required to treat certain human diseases and can contain biological products, but such products are often expensive to make, and the transgenic animals that produce the useful biological products can be created by the introduction of the portion of the DNA or genes which code for a particular product, such as the human protein alpha-1 antitrypsin, and used to treat the emphysema, and the similar attempts have been made for the treatment of the phenylketonuria and the cystic fibrosis. In 1997, the first transgenic cow Rosie was produced the human protein enriched milk, and the 2.4 grams per liter of the milk contained the human alpha-lactal women, and was nutritionally a more balanced product for the human babies than natural cow milk. And the vaccine safety and the transgenic mice are being developed for use in testing the safety of the vaccine before they are used on humans. And transgenic mice are being used to test the safety of the polio vaccine. It's successful. We want to be reliable and could the use of the monkeys to test the safety of the batches of the vaccine, the chemical safety testing, and this is the toxicity of safety testing, and the procedure is the same as that of the use of the testing. Toxicity of the transgenic animals are made that carry genes which make them more sensitive to toxic substances than the non-transgenic animals when they are exposed to the toxic substance, and then the effects are studied in the toxicity testing in such animals will allow us to obtain the results in less time. The physical issues, the manipulation of the living organism by the human race cannot go any further without regulation. And some ethical standards are required to regulate the mortality of all human activities that might help or harm the living organism. Going beyond the modality of such issues, the biological significance of such things is also important. The genetic modification of the organism can have unpredictable results when such organisms are introduced into the ecosystem, and therefore the Indian government has set up organizations such as the GEAC, Genetic Engineering Approval Committee, in which will make the decisions regarding the validity of the GM research and the safety of the introducing the GM organisms for public services. The modification or uses of the living organisms for public service as the food and medicine resource, and for example, has created problems with patients granted for the same. And there is a growing public anger. The certain companies are being granted patents for products in the technology that make use of engineering materials and the plants and other biological processes that have long been identified, developed, and used by the farmers and indigenous people of a specific region of country. And the rice is an important food grain, and the presence of which goes back to thousands of years in the Asia's agricultural history. And there is an estimated 2 lakh varieties of rice in India alone, and the diversity of the rice in India is one of the richest in the world. The basmati rice is distinct for its unique aroma and the flavor, and 27 documented varieties of basmati rice are grown in India, and there is a reference to basmati rice in ancient texts and the folklore and poetry, and it has been grown for centuries. In 1997, an American company got rights on trademark got patent rights on the basmati rice through the US Patent and Trademark Office. And this allows the company to sell a new variety of the basmati in the US and abroad, and the new variety of the basmati has actually been derived from the Indian farmers' varieties, and the Indian basmati cross the patent dissolved to non-functional equivalence, implying that the other people, the new variety of the basmati was actually being derived from the farmers' varieties, and the Indian basmati was crossed with the semi-raw varieties that has claimed as an innovation or novelty, and the patent extends to functional equivalence, implying that other people selling basmati rice would be restricted by the patent. And several attempts have also been made to the patent uses products and the process and the based on the Indian traditional herbal medicines and the remedic name. If you are not vigilant, we do not immediately occur this immediately counter the patent application. The other countries and individuals may encash our rich legacy, and we may not be able to do anything with it. And bioenergy is the term used to refer to the use of the bioresources by multinational companies and other organizations without the proper authorization from the countries and the people concerned, without the compensatory payment. And the most of the industrialized nations are financially rich but poor in biodiversity and traditional knowledge, and in contrast, the developing and the underdeveloped world is rich in the biodiversity and traditional knowledge related to bioresources, and the traditional knowledge related to bioresources can be exploited to develop the modern application and can also be used to save time, effort, and expenditure during the commercialization. And there has been the growing relation of the industries in adequate and compensation and benefit sharing between the developed and the developing countries, and therefore some nations are developing laws to prevent such unauthorized exploitation of the bioresources and traditional knowledge. And the Indian Parliament has recently declared the second amendment of the Indian Patents Bill that takes such issues into consideration, including the patent term, emergency condition, and research and development initiative.